Hello, my name is Holly Huse. I am a postdoctoral fellow at the University of California, Los Angeles. Welcome to this Pearl of Laboratory Medicine on laboratory detection of carbapenem resistance in gram-negative bacteria. Here are the objectives for today's presentation. I will review the mode of action of beta-lactam antibiotics, describe different classes and subclasses of beta-lactam antibiotics, review beta-lactam resistance mechanisms and classes of beta-lactamases, discuss methods for detecting carbapenem resistance and indications for carbapenemase testing in gram-negative bacteria, and explain methods for detecting carbapenemases. Pathogenic gram-negative rods are broadly divided into the Enterobacteriaceae and glucose nonfermenters. The Enterobacteriaceae are colonizers of human and animal gastrointestinal tracts, or are environmental. Genera within this family include Escherichia, Shigella, Citrobacter, Salmonella, Edwardsiella, Klebsiella, Enterobacter, Serratia, Proteus, Providencia, Morganella, and Yersinia. Glucose nonfermenters are environmental and cause opportunistic infections. The most commonly encountered nonfermenters are Pseudomonas aeruginosa, Acinetobacter species, and Stenotrophomonas multifila. Both groups cause diverse infections, including pneumonia, wound or surgical site infections, meningitis, gastrointestinal infections, and bloodstream infections. Beta-lactam antibiotics are often used to treat infections caused by gram-negative rods. Beta-lactams share a common core structure, the beta-lactam ring, and are grouped based on structural similarities. Penicillins include amoxicillin and ampicillin and have the narrowest spectrum of activity. Beta-lactam inhibitor combinations have a beta-lactam and beta-lactamase inhibitor and include amoxicillin clavulanate, ampicillin sulbactam, piperacillin tazobactam, ceftolozane tazobactam, and ceftazidime avibactam. Cephalosporins are further divided into generations, with higher generations having greater activity against Enterobacteriaceae. The cephomycins, cefoxitin and cefotetin, are similar to cephalosporins. Aztreonam is the only available monobactam and has activity against aerobic gram-negative bacteria. The carbapenems, doripenem, ertapenem, imipenem, and meropenem, are some of the most important beta-lactams because they have the broadest spectrum of activity. Often considered last resort antibiotics, they are reserved for serious infections. Carbapenem-resistant Enterobacteriaceae, or CRE, are usually resistant to all beta-lactams, severely limiting treatment options for patients with these infections. Beta-lactam antibiotics target bacterial cell wall biosynthesis. They bind to and inhibit penicillin-binding proteins, or PBPs, which are required for building peptidoglycan, an essential cell wall component. In gram-negative bacteria, beta-lactams enter the periplasm through outer membrane porin channels and bind to PBPs, causing cell death. Beta-lactam resistance can be conferred by beta-lactamases, enzymes that hydrolyze beta-lactams. There are many different types of beta-lactamases with varying capacities to hydrolyze beta-lactams. Carbapenem resistance can arise via two mechanisms: bacteria with an extended spectrum beta-lactamase, or ESBL, or an AMP-C beta-lactamase can poorly hydrolyze carbapenems. However, in combination with porin loss or obstruction, these enzymes can confer carbapenem resistance. These organisms are non-carbapenemase producers, or non-CP. A bacterium can also acquire a carbapenemase, which efficiently hydrolyzes carbapenems and confers resistance to carbapenems. These organisms are carbapenemase-producing organisms, or CPOs. Enterobacteriaceae are carbapenemase-producing CRE, or CPCRE. Differentiating between these mechanisms is important because carbapenemases are transmissible and therefore important for infection control. CP-CRE can hydrolyze penicillins, cephalosporins, monobactams, and carbapenems. Ampler classification divides beta-lactamases into four classes via amino acid sequence homology. In this table, carbapenemases are shown in red. Class A beta-lactamases include ESBLs and KPC and are found in the Enterobacteriaceae. KPC is the most common carbapenemase and was discovered in Klebsiella pneumoniae. These enzymes are endemic in the United States. Metallobeta-lactamases are Class B enzymes and include the NDM, VIM, and IMP carbapenemases. Unlike Class A, C, and D enzymes, which contain active site serines, Class B enzymes contain zinc. Class B enzymes are found in Enterobacteriaceae, P. aeruginosa, A. baumannii, and S. multifolium. These enzymes are endemic in India and Asia and are becoming more common in the United States. AMP-C beta-lactamases are Class C enzymes. They are found in some Enterobacteriaceae and non-fermenters. Expression of AMP-C beta-lactamases is inducible or constitutive. Class D contains the oxocarbapenemases, which are found in A. baumannii and the Enterobacteriaceae. They hydrolyze carbapenems and are endemic in Asia and Europe. The Clinical Laboratory Standards Institute, or CLSI, outlines methods for laboratory detection of carbapenem resistance in their M100 S27 document, Performance Standards for Antimicrobial Susceptibility Testing. Methods include disk diffusion and broth microdilution. In June 2010, CLSI lowered the Enterobacteriaceae minimum inhibitory concentration, or MIC, breakpoints for carbapenems. The MIC is the minimum concentration of antibiotic required to inhibit growth of an organism. The current interpretive categories and breakpoints for Enterobacteriaceae are shown. Isolates are susceptible to doripenem, imipenem, or meropenem at an MIC less than or equal to 1 microgram per mil, intermediate at an MIC of 2 micrograms per mil, and resistant at an MIC greater than or equal to 4. For ertapenem, isolates are susceptible at an MIC less than or equal to 0.5 micrograms per mil, intermediate at an MIC of 1 microgram per mil, and resistant at an MIC greater than or equal to 2 micrograms per mil. MIC breakpoints were revised based on evaluation of pharmacokinetic, pharmacodynamic properties, clinical data, and MIC data that included the evaluation of CPOs. When using the current MIC breakpoints, routine carbapenemase testing is not required. Disk diffusion breakpoints are not shown. When using the current breakpoints, perform antibiotic susceptibility testing on isolates of Enterobacteriaceae and interpret the results based on the current breakpoints. Report the results for treatment purposes and perform carbapenemase testing only for infection control or epidemiological purposes. Perform carbapenemase testing on isolates that test intermediate or resistant to one or more carbapenems. When using the old MIC breakpoints, perform carbapenemase testing on isolates of Enterobacteriaceae with an ertapenem MIC of 2 micrograms per mil or imipenem or meropenem MICs of 2 to 4 micrograms per mil. CLSI recommends using the CarbaNP, the Modified Carbapenem Inactivation Method, or MCIM, or molecular testing for detecting carbapenemases. An asterisk appears next to the Modified Hodge test because while current recommendations describe using the Modified Hodge test for carbapenemase detection, this recommendation is changing and CLSI will no longer recommend this test. Use any of these methods to test for carbapenemases in Enterobacteriaceae. Use the CarbaNP or molecular methods for testing Pseudomonas aeruginosa and Acinetobacter species. For phenotypic tests, report positive results as carbapenemase positive or carbapenemase producer and negative results as carbapenemase not detected. While the modified Hodge test will no longer be recommended by CLSI, I will describe it here. This test measures growth of a susceptible E. coli strain in the presence of a carbapenem disc and a potential carbapenemase producer. A lawn of susceptible indicator E. coli is spread on a Mueller-Hinton agar plate, and a 10 microgram disc of ertapenem or meropenem is placed on the lawn. The test isolate is inoculated in a straight line approximately 20 to 25 millimeters in length from the disc edge, and the plate is incubated 16 to 20 hours. If the test isolate is a carbapenemase producer, the carbapenemase will diffuse into the agar and hydrolyze the antibiotic, allowing growth of susceptible E. coli around the test organism and towards the carbapenem disc, as shown in the bottom panel for isolates 5 to 8. This results in an indentation of growth towards the carbapenem disc due to carbapenemase production by the test organism. If the organism does not produce a carbapenemase, E. coli growth is not indented toward the carbapenem disc, as shown in the top panel for isolates 1 to 4. The advantages to the modified Hodge test are that it is simple to perform and no special reagents or media are required. However, false positive results can occur in isolates that produce an ESBL or AMP-C coupled with a poor mutation. The test has poor sensitivity for the New Delhi metallo-beta-lactamase, as shown. The CarbaNP is a colorimetric test for carbapenemases. The test isolate is emulsified in a microcentrifuge tube containing extraction reagent. Solutions containing imipenem and a pH indicator are added, and the reaction is incubated for up to 2 hours. If a carbapenemase is present, the imipenem is hydrolyzed, decreasing the reaction pH and leading to a color change from red or red-orange to light orange, dark yellow, or yellow. A negative reaction remains red-orange. An invalid reaction changes from red or red-orange to orange. The image shows a negative reaction and a positive reaction for a KPC producer. One advantage to the Carba-MP test is that results are obtained in 2 hours or less. Positive tests are reported as soon as a color change occurs. This test can be performed on Enterobacteriaceae, P. aeruginosa, and Acinetobacter species. Limitations to this test are that some reagents require in-house preparation and have short shelf lives. Colors can be hard to interpret, invalidating results, and this test has low sensitivity for oxotype carbapenemases as shown. The MSIM test is a carbapenem inactivation test. The test isolate is suspended in tryptic soy broth in a microcentrifuge tube, and a 10 microgram disc of meropenem is added to the suspension. The tube is incubated for 4 hours. A Mueller-Hinton agar plate is inoculated with a lawn of indicator E. coli. The meropenem disc is removed from the reaction tube and placed on the lawn, and the plate is incubated for 18 to 24 hours. If the test isolate is a carbapenemase producer, the enzyme will hydrolyze the meropenem within the disc, and there will be no or limited inhibition of E. coli growth. The reaction is positive if the zone of inhibition is 6 to 15 millimeters, or colonies are present within a 16 to 18 millimeter zone. In the figure, the positive test shows no growth inhibition of E. coli. A negative reaction has a zone size that is greater than or equal to 19 millimeters, as shown. The test is indeterminate if the zone is 16 to 18 millimeters. The advantages to the MSIM test are that no special reagents or media are required, and it is highly sensitive and specific. Additionally, CLSI now endorses this test for P. aeruginosa. However, the MSIM requires overnight incubation, increasing time to results, and not all carbapenemase-producing Enterobacteriaceae are MSIM positive. For example, one isolate of OXA-232 producing Klebsiella pneumoniae was negative by this assay at some validation sites. Molecular testing can be performed on Enterobacteriaceae, P. aeruginosa, and Acinetobacter species. It determines the presence or type of carbapenemase. The only FDA-cleared test is the Cepheid Expert Carba-R, which detects KPC, NDM, VIM, IMP1, and OXA48. Advantages to molecular testing are that it is rapid and can distinguish different carbapenemase types. However, false negatives can occur if the test isolate has a carbapenemase not targeted by the assay, and it is more expensive because special reagents and equipment are required. If your lab is using the current carbapenem breakpoints, perform carbapenemase testing if requested by infection control or for epidemiological purposes. If your lab is using the old carbapenem breakpoints, carbapenemase testing is required in isolates that have an ertapenem MIC of 2 micrograms per mil or imipenem or meropenem MICs of 2 to 4 micrograms per mil. Tests endorsed by CLSI for carbapenemase testing are the Carba-MP, MSIM, and molecular testing. Remember that while the MHT is in current guidelines, it will no longer be recommended by CLSI. Challenges to carbapenemase testing are that ertapenem resistance is often due to ANSI or ESBL. Proteus providentia and Morganella have intrinsically elevated imipenem MICs, and no phenotypic test detects all carbapenemase resistance mechanisms. Thank you for joining me on this Pearl of Laboratory Medicine on laboratory detection of carbapenem resistance in gram-negative bacteria.